I have a real surprise for you today. And Linda Kelly is going to come up and talk to us about viruses. And I think this is going to be one of these aha moments for you. This is going to kind of be a continuation of actually the first PERF lecture on bacterial pathogenesis. And do we have any postgrads in the room? No postgrads? One postgrad, two postgrads. So Linda comes from a lab in San Diego that is known the world around for doing Sanger sequencing on bacterial viruses. And they've come up with some very, very interesting information on what really makes bacteria pathogenic. Something that's been completely off our radar screen. For example, does anyone know the cause of cholera? Can someone here tell me what causes cholera? Vibrio cholera, right? Are you sure? So is Vibrio cholera both necessary and sufficient to cause the disease? Certain other criteria. Okay, so Vibrio cholera is not both necessary and sufficient for the disease to occur. Is that correct? Yeah. Okay, so I want you to give a warm welcome to Linda. The reason I had her come here is next year's scientific session is from Forrest Roher, the lab that she works in, and she's going to rock your world. Linda?
Thanks. No pressure or anything. Thank you, guys. Well, Gary asked me to come here to get you excited about Phage, and so that's what I'm going to attempt to do. So thank you. My name is Linda, again, and I work at, I have an adjunct position at SDSU. Okay, so phage are viruses, but they don't infect your own tissues like a cold virus, for example. Viruses infect bacteria or archaea. They're both prokaryotes. You're all familiar with bacteria. I don't know how many of you know what an archaea is. It's like, it is, so archaea are small like bacteria. They're single-celled organisms. They're as different from bacteria as you are from bacteria. They're basically that divergent. And they're most well known for living in like boiling springs, like hot springs, low acidic, high salt environments. So these phage will infect both bacteria and archaea. And so they're obligate parasites. In order to complete replication, they need to infect a host. In this case, I'm just going to talk about bacteria. They're going to infect a bacteria. They're going to utilize all that bacteria's machinery to replicate its genome to build new particles. And then they're going to blow up that bacteria ultimately. Okay. In some cases, I'm going to show you, they have small genomes that need to be packaged in these little phage heads. Okay. So their whole genome's in here. And so they can keep their genome relatively small because they're using all the machinery of the bacteria. They don't need to encode all these genes to do everything. They just need the ones that they need. And so they always have this, so they have nucleic acid here, and then they're always coded, this is all protein. That's called a phage capsid. And this is just a little cartoon to introduce their life cycle. I'm going to go through it a couple of times. But so what they need to do is they need to find a bacteria. They need to bind to it, inject their own genome, make copies of themselves, and then release themselves out of the bacteria. Right. This is how many viruses there are in the world at any given moment. Okay, that's a lot of zeros. It's 10 to the 31. And to be honest, like that, it is a lot of zeros, but it doesn't really mean a lot to me, 10 to the 31 phage in the world at any given time. They are the most abundant entity on the planet, whereas so bacteria would be 10 to the 30, and there are about 10 times more phage than the bacteria on the planet. So that's in the ocean, in the soils, in the sediments, in the deep biosphere, in the air. And so because this didn't really seem like a lot, and it is a lot, there's a really cool article in the Washington Post that said if you lined phage up head to tail in a little parade of phage, they'd extend out to space to 200 million light years. Well, that seems pretty far, right? Given that they're so small. And that is 80 times further than the farthest object we can see with the most powerful telescope. There's a lot. And so if you hate bacteria, then you should really love the viruses that blow them up. This is an epifluorescent micrograph of, this is seawater. This is about a drop of seawater. And here, these big white blotches, those are your bacteria. And all these little things that look like stars are viruses. Phage, namely phage, okay? And so since I've been talking, on Earth, there's been about 10 million metric tons of bacteria blown up by phage. There's a lot of work happening, okay? Right. So phage are predators. They should only be known as predators, right? They're obligate parasites, they're predators. If that were you, I don't know what you'd be thinking. But if you were a bacteria and that were you, you'd probably both be thinking the same thing. You're going to die very soon. We have many ways to study viruses and phage now. You can culture them on a plate. And so all of that kind of opaque, that's a bacterial lawn. I'm sure many of you have seen that. And if you just drop liquid that has phage in it that infect that bacteria, you'll get this clearing, and we call them plaques. It's basically a hole in the lawn of bacteria, and that's from the phage lysis. So just infected a cell, lysed it over and over until you get into a position where you can see a clearing of bacterial cells, okay? So just the same way that you can culture only a small amount of bacteria, you can only culture a small amount of viruses or phage that are on Earth. You can also count them directly, and so this is another epifluorescent micrograph. And so this is seawater, and I've literally counted thousands of samples that look like this. It's not the worst way to spend your time, but it does get a little old. Okay. But so, and what we do is we look at all these bacterial and phage dynamics, like in a system, how many bacteria there are, how many viruses, and we go from, I mostly focus on tropical marine environments like coral reefs. So we go in, we scoop up some seawater, we look at what's going on. We can count how many of the things are there using these direct counts. We do a lot of sequencing, which tells you who is there and how many of each type of bacteria or virus are there. And so you can generate these graphs of what types of phages are in an environment. So these are the phage capsids, right? These are protein building blocks that basically protect the most important part of the phage, which is its nucleic acid, its genome. And so they tend to be very intricate and detailed looking proteins, and so they are the subject of a lot of artists' work, both sculpture and drawing. And so I'm going to show you a lot of pictures. This is all kind of phage art, but this is what they look like. They have different shapes. So we have a phage with very long tails. This is a lambda phage, so it affects E. coli. And then so this would be the phage head, and the tail, that's all capsid, okay? And then this is like your very stereotypical lunar lander looking T4 phage, the one you've probably seen pictures of. And then you even have phages with really tiny short tails. Sometimes you can't even see them. You can only really see the phage head. And so the capsid's role is to protect the genome. This is in the environment. They can be in water, soil, salty water, hot water, acidic water. And this protein structure protects them, even in the gut, right? It's got to get through your stomach to get into your gut, things like that. And what also, the way they're designed is, so this is called an icosahedral shape. And what it is it's 20 equilateral triangles. And what that means is that you only have to encode one shape. So in your genome, so you make one triangle, and then you put 20 of them together, and you get this beautiful ball. And then also, these are just like protein subunits that are all the same, and then they make the tail. And so it allows them to keep their genome small, which is beneficial to them. So there's just a couple, and I know this is kind of like a lot of really dry, boring details, but I'm going to tell you a couple stories in the end, and all of these body parts are going to come into play. So you got to remember them, or else you won't understand the story. So you have this tail sheath that helps it for the injection of the DNA into the bacterial host, and then these tail fibers are actually what recognize specific structures on the bacterial host that allow them to bind and attach and to invade the host. So this is more phage art. It's like beads. There's a lot of it. You can check it out online. Okay. So they have two basic lifestyles that I'm going to talk about. The first one's the lytic lifestyle, which is what I'd shown you, where it essentially ends with blowing up the bacteria. You have to first find a specific region on the bacterial cell. You have to bind to it, inject your genome, replicate, and then assemble virions with all those capsid proteins. And then you got to find a way to get out, right? You're locked inside this intact bacterial cell. You have to literally blow it up from the inside out so that you can get out and go and infect a new cell. The name of the game is to copy yourself, be the most abundant phage. So I'm going to just hone in on two of the parts of this lifestyle because they come into play later. These tail fibers recognize specific structures that I talked about. Generally, they bind to receptors. These are the way bacteria actually absorb the nutrients that they need to survive. This is how they import carbon, sugar, the Snicker bars that you ate that are now feeding all the bacteria in your mouth, right? This is how bacteria don't have a mouth. What they have is receptors all along the outside, and they kind of absorb everything through these transporters. That is what a phage finds and recognizes, and that's how it gets in. Generally, it's a receptor like that, sugar receptor. And so phage do tend to be, there are more generalist phage that can kind of go, especially within a species of bacteria, but maybe even between some species. But some phage are so specific that it's at strain level. And sometimes they even use phage to identify what strain of bacteria is because it can be so host specific. This is just a drawing of the lifestyle, the life cycle, the lytic life cycle. And what I want just to illustrate here is that the number of virions that you build inside the cell is called burst size, and that can be important just with kind of the ecology of bacteria and phage dynamics, is that sometimes maybe you make 10, the burst size of 10, other times it's like 100. Okay? And so it kind of, and that really comes into play with like how prolific you can be in killing a bacterial community, or how fast. And so this is an electron micrograph of basically a cell bursting. And so this would be, you can see the phage inside the cell, and that the structure of the cell has been compromised right here, and the virions, the phage, are coming out. It almost kind of looks like a balloon popping, if you put it, kind of everything coming out. And so there are two of these proteins are going to be important later, okay? These two proteins that help with it. These holins are proteins that the phage makes. The phage encodes it on its own genome, and it pokes holes in the cell membrane of the bacteria. Okay? Through these big holes, they're kind of like a big porin protein, like a beta barrel. Then there's other proteins called lysins, and those break up the cell wall, and that is actually what kind of makes the balloon pop. And I'm going to show you a video of lysins. You can, these lysins have been sequenced, and they can be made in the lab, and then you can just drop lysins onto bacteria and watch them blow. And I'm going to show you a video of that. Okay. So happy bacteria, gone bacteria. You can't even see anything left. Just gone. This is Bacillus anthracis. They're nice to look at because they're giant. Those are the little, they cause anthrax, those little bastards. All right, and then another video, which is kind of an artistic video, but it's going to give you a break from me, and it's going to give me a break to drink water. And it's cool. It's showing the lytic cycle. It has a little bit of sound, but it's kind of just funky music. I don't know if you need to hear it. Hey, Felix, can you do the sound just a tiny bit? There it is. So this was drawn. This isn't. And that's actually true. The bacteria does fill up entirely. If you catch one at the end, it's like completely filled with virions. I don't know where all the other stuff is. Well, so you can do it in a lab, like with an E. coli strain, and it cycles about every 20 minutes, but probably in the environment, more like a day. But it also kind of demonstrates that if you have a lot of the same cells around, like how this whole lytic cycle would work. Like if you have a lot of bacteria, and you have a lot of phage, you're getting a lot of them out, and then they're going and infecting somebody new. So, you know, you can make lysates that are like, you can generate like 1 billion viruses or something in the lab. All right, so that is how the phage acts as a predator and kills bacteria to propagate itself. And so in so doing this, they're really changing the communities that are there because they are selecting for certain bacteria. Okay? And so if the phage kills all of that bacteria, then it isn't there anymore. So a lot of the diversity and changing of populations with bacteria are driven by this predation. Okay? That's really important. And in the environment, like in the ocean, for example, you're also releasing a ton of carbon back into the environment when you blow up the bacteria. So here you see these are the baby virions coming out of the cell. But all of this that you see right here, like the cytoplasm, that's all nutrients that can go back into the system for other bacteria to eat. So nitrogen, phosphorus, carbon. So when they're blowing up these cells, they're they're actually providing more food to other cells in the environment. And that's important. They're also structuring the community. And then this dynamic is what we call kill the winner, where if a strain of bacteria becomes particularly dominant in an environment can be not just like an outside environment, but like in your mouth, right? That a virus will come in and kill that bacteria, okay, and somebody comes in their place. Okay? And so what you see, if you were just to take like that picture that I show you, an epifluorescent micrograph, and you counted the number of bacteria, there would always be about 1 million, if you're in the ocean, per drop. Okay? And there will always be about 10 times that many viruses there as well. But who is there is changing a lot, like this. So you have this one bacteria, it's growing, it's very happy. Once it gets to a high concentration, a virus that's specific to it comes in, and here it comes up, the same time that bacteria goes down because it's killed it, okay? And so now and then it released all this food, and so now there's another bacteria that was in a low abundance, and now it's like, hey, I've got nice food to grow. It grows up, and once it gets pretty abundant, now another virus that infects it can find it. It's basically just the chances that it runs into each other, and it's going to knock it down, okay? That's happening all the time in the environment, in the body. And so this is what we think about it more like at the strain level that you're changing. So even if you're collecting a sample and you always see that you have, 30% strep, well, that's fine. But you don't have the same strep all the time. Okay? If you have 30% strep all the time, you take a sample every day from a person, it's like here. Okay? But this is what's happening at the strain level. Okay? It's always changing, okay? And they're a little bit different. This one, maybe it was only 10%. It's always about at 10%. But you have these little ones going. Okay? This is very common. This is the way it works. And so the way we think about it is this evolutionary model that we call the Red Queen, and it's named after Lewis Carroll's Through the Looking Glass, where the Red Queen has to run faster and faster in order to stay where she is. That's what we're all doing. This is like the evolutionary arms race between predators and prey, where you evolve to escape the predator, and that predator evolves to get you back. Okay. We see this all the time in medicine. Right? Okay, so I'm going to talk, in that regard, this is related to 2 ways that bacteria go about evading the predator, the phage. What are their defenses? Very related to that evolutionary arms race is that they change their cell receptors so that they could evade predation by a phage. If you have a phage that's binding to a certain sugar receptor on your cell wall that you use to import nutrients, if you change it a little bit, that phage can no longer bind to it. And so you're going to evade that predator until it evolves to be able to infect you through that receptor again. Okay, so I'm going to show you that. And then there's a second mechanism I'm going to talk about called CRISPRs, and it's a really cool system. It's actually kind of like a miniature bacterial adaptive immune system. So to illustrate the receptor story, I'm just going to show you a story, a study from our lab where we cultured roseophage. So they infect Roseobacter, which is a common marine bacteria. And so just like with the plates that I showed you with the plaques, this is how we isolated those from here. We would grow Roseobacter on a lawn, and we would just drop seawater on it, and we would take plaques. And what we found is we found 5 different phages from different places and different years, and we sequenced their genomes. The phage, not the Roseobacter. And so these are, this is basically what their genomes look like. So they were very similar, obviously. So 96 to 99% similar between the 5 phages that we looked at. That's all in this pink. But so I want to draw your attention to this region where you have this white. That is divergence in the genome from one another. And what they are is this is their tail protein. So they're changing their tail protein because that's what they need to recognize the bacteria, and the bacteria is changing it. This is this red queen dynamic that we think about. And so these are some examples of the cell receptors that phage can bind to. And these are all like nutrient transporters, like I talked about. They can even bind to flagellar protein that's on the cell. Okay. And so when the bacteria, if they, like sometimes they just bring them in. They're like, well, I don't need this transporter right now. It's a receptor for phage. I'm just going to bring it in so it can't bind. Okay. Or they somehow change it. But the phage in turn changed their tail. It's almost like an antibody system, right? It's like a lock and a key. Got to get the right fit so that you can inject the genome.
Are these just spontaneous mutations that are selected for? Or?
Yeah, they're selected for because you're not being killed. But yeah.
The mutation is just a regular spontaneous mutation in the gene that allows it to survive.
It would. It's like genetic drift. You'd be getting mutations, and they would work to your benefit. Yeah. And so then I'm going to introduce CRISPRs. And the CRISPR system's a little bit complicated, but I'm just going to explain the whole story and see how it goes. Okay. This is the region of the genome that we call the CRISPR-Cas system in the bacterial cell. This is what it looks like. It has a number of these Cas genes that are proteins that are made by the bacteria that do stuff. Okay. This leader sequence is where they're packaging pieces of the phage genome, or at least exact copies of the phage genome that came into the cell. And then the reason they were found is because they have these so CRISPR stands for clustered, regularly interspaced palindronic repeats, which is what were found first. And I'm like, what is going on? You've got these weird palindrome regions, and palindrome's like a word like kayak, reads the same forward and backwards. You have a lot of DNA sequences that do that are palindromic, okay? So they had these palindrome repeats, which are in gray, and then these random sequences that didn't match anything in between. These random sequences match phages that had infected this host. And so what's important to know, and that what this picture doesn't illustrate very well, is that these get packaged based on some kind of a failure of a phage infection. If it was a successful phage infection, that bacteria would be dead and gone, okay? Somehow you're getting some kind of infection and it can't go to completion. In the time that they had the genome in there, these Cas proteins are taking a copy of some of that foreign DNA, and they're putting it right here, and they put it closest to the leader. So the most recent virus that you got a little copy of and you put in your genome is here. And then along the array, they keep putting the new one here, and it's only so long, so the old ones actually fall off. You haven't seen it in a long time. Eventually it just goes away. You don't have it anymore. If you have it, it's a recent virus that's been here. If you get infected by a new virus, you will have these little complexes that these are, it's base complementary to what the genome's going to be like. It will bind to, it'll recognize a certain fragment of the genome, bind to it, and it's going to tell the cell to cut it up. It's going to destroy it. So it's a protection. And it's like an adaptive immune system for bacteria. It's incredible, right? They're so simple.
Do all bacteria have this?
I don't think all bacteria do. Yeah. But many of them do. Okay. This is relatively new. 10 years maybe. What time is it, Gary? Oh, okay, we're good. Okay. So we talked a lot about the lytic life cycle, just infecting cells, blowing them up. Another way that a phage lives with a bacteria is kind of more symbiotic. Instead of just going in, replicating, blowing it up, it incorporates its genome in the bacterial host genome, and then it just divides with the bacteria as it divides. And so there's kind of a lot of arguments about why a phage would do that. But one reason would be is that if you thought you were if you thought you were unlikely to find another host nearby, there doesn't seem to be another one, that you just wait it out. So you go in there and it's growing. When conditions get really good for that bacteria, it has a lot of copies of itself, then you go out and then it's easy to find another host. Okay. That's one reason to do it. Okay. But this really affects biology, this lysogenic cycle, in a lot of ways, because now you just added new genes to a genome. Okay? And so that's what I'm going to spend most of the rest of, and the stories I tell you, they're all related to this lifestyle. And so this is another drawing, and I wish that they would have, the only thing I wish they would have done is made this colorful. Is that this is the bacterial genome here, and you can't really see that the phage genome's been inserted in there, but it is. You just got to imagine that this is like a little pink place, or blue, and that's where the virus has inserted its genome into the host genome. And like I mentioned, they can become, so you have the genome incorporated, it'll get a signal that there's like a lot of food out there or a lot of new hosts, and it will induce itself. It'll pop back out of the bacterial genome. It'll start making copies of its own genome and start making the proteins to build capsids and go through the lytic cycle. And so then I'm going to just introduce these terms because if I say them later on, I want you to kind of know what's going on. So this is the lysogenic cycle is when this integration has occurred. A phage that has done that integration is now called a prophage. So you'd say that this bacteria has a prophage, which means it has this foreign virus in its genome. Induction is when the virus gets some signal to pop back out. And we say it becomes induced, and that means it's basically removed that piece of nucleic acid out of the genome, and it's going to become a virion again. And just to make it more confusing, lysogenic phages are also called temperate phages, which means they can decide to be lysogen or a lytic phase or a lytic phage. And I don't really know how you'd be a lysogenic phage if you never went lytic, but that's the term. And it's gonna, and I just wanted to introduce it because they're kind of used interchangeably, temperate and lysogenic. And this is just to show you, so they don't just like pop into the genome anywhere. They just like get in and they're like, oh, that looks like a good spot, because that would be a terrible thing for the bacteria if you just like shift everything in the whole genome. That's not going to work right anymore. So what they have is they have specific sites in the bacteria. They're called at sites. So if this is a E. coli genome, this at B is this region on the bacterial genome that recognizes at P, that's on the phage genome. They actually bind to one another and they do recombination, and that's how you get it inside. And so then you also get excision, it pops out. And so it kind of shows it here where they meet. But here, if you can see, and it's not super clear is that they should have put a circle, a half circle, and a half square or something, because really it's cut right down the middle, and it matches with the left side here. That's here. And the other half here and here is here. And this is the phage genome. See, it's backwards because it recombined. It's here. Phage. Okay. And so what this means is that the phage can bring in genes that can do anything, namely virulence factors, right? Now you can be a phage that comes in and gives some special powers to this bacteria that ultimately the reason why you do it from a phage perspective is if it helps with proliferation, right? If you can make more bacteria, then you can make more phage particles. So these are a list of bacteria here. These are the diseases that they cause. And these diseases literally would not exist if they didn't have a lysogenic phage. The phage is what, and in many cases, they're toxins. So here, particularly, and I'm going to give you an example. I'm going to show you the Vibrio example, but also Shigella. You know, you have tons of E. coli in your gut. You don't have Shigella. The E. coli that you get, if you got Shigella, would have a lysogenic phage in it that's carrying the Shiga toxin. And that is what causes all the damage to your cells. Shiga toxin basically puts a big giant pore in your cells and just like blows them up. E. coli can't do that on its own. Botulism, diphtheria. If there's no phage, there's no pathogen, okay?
So what causes cholera?
We're going to talk about cholera. It's true. So it isn't just Vibrio cholera. There's Vibrio cholera all over the ocean. Nobody's getting cholera from swallowing it. There's this very complicated things that have to happen for cholera to happen, okay? So cholera, everybody knows that it's caused by Vibrio cholerae. What causes the disease is this cholera toxin that is what causes the diarrhea. And the toxin is carried on the phage, which I just showed you. And there it is, temperate. See, I told you. Be confused if I didn't give you that. So this is how it goes. We just have this very nice and friendly Vibrio cholera in the ocean. It can't cause cholera. It's just living there happily. At least two things has to happen for that Vibrio to be able to cause cholera. The first thing it has to do is it has to get this TCP island, which stands for toxin co-regulated pilus. It's probably one of the things that they found was a virulence factor for cholera. And what it does is that it makes this pilus that's on the outside, this kind of a filament that's on the outside of the cell called TCP. If you don't have any TCP, you don't have any disease. This is also kind of unknown because this comes as a mobile element, and some people think that this is also a phage. You get a first phage that comes into the cell. It goes lysogenic, and now you make this pilus, filament, okay? This filament is the receptor for the phage that carries cholera toxin. So if you don't have the filament, you don't have a way to get in. The cholera toxin carrying phage can't infect the cell, okay? That's amazing, right? How did that even happen? So here you go. Now you have the cholera toxin phage. It finds the filament. It attaches with its specific receptors. It infects the cell. It incorporates its genome into the DNA, and it just sits there. It doesn't get induced when it causes a disease. It's living there as a temperate phage, and this is your cholera-causing Vibrio. Okay. Once it's inside, it's carrying this cholera toxin, that gets made by the bacteria machinery anyway. It's just like a normal gene that's inside the genome now, and it gets made. The proteins get made inside the cell. The cholera toxin is a typical, we call these alpha-beta toxins. With these types of toxins, the beta subunit is always the piece that binds to the cell. In this case, it's epithelial cells in your gut. So it binds to the surface, and then the alpha subunit is like the active site. That's what you call. So what happens is that the whole thing gets pulled into the cell. This is an epithelial cell. They look like this, right, in your gut. I know you guys don't study guts, but they all went to medical school, right? Here's your Vibrio cholera. They're growing on the outside. They're releasing toxins outside the cell. Toxins are binding all along here. They're getting endocytosed. They get processed by the ER. And then this little piece, which is the alpha subunit, it starts messing with cellular metabolism. What it does, how it messes with cellular metabolism is it makes you start pumping ions across these channels, these ion channels, so sodium, potassium. When you pump out ions, water comes with them. It just has to. Okay. The ones in the front? I guess that's the backlighting. There you go. Okay. So see, we have like sodium, potassium being effluxed out of the cell through these ion channels. That makes all the water from inside the cell come out. And so the water's coming out here into your gut, and then it's coming out, right? So that's what causes the diarrhea. I mean, people with cholera lose like liters of water a day. They don't die, they die from dehydration exclusively. You don't die from any damage to your cells or anything, okay? And so why you would do that if you're a bacteria or a phage? Well, you're growing, water's coming out, you're being washed out, into the river, and now you're going to new hosts. And so you're making like tons and tons, and actually the titer of Vibrio cholera is huge, enormous. So all those viruses, all that bacteria, and none of that would happen if you didn't have the virus. So self-propagation. All right, that's story one. All right, story two is an oral microbiota story just for you guys. Okay, so we did a study in our lab where we took 20 healthy humans, and this was the reason we did the study was to see whether you could monitor populations for certain viruses. Okay. But we learned a lot of other things by doing the study. So what we did is we pooled 20 individuals from these oral pharyngeal samples. They were swabs. And we sequenced the viruses that were associated with these samples. And so this was the population of bacteriophages that we found in this pool of samples, so remember. And so the pool is kind of good because if you have a different population and a lot of different people, the pool is kind of nice because it kind of gives you like a bigger view of what could be, I suppose. It was also because in those days, sequencing was a lot more expensive. So we're going to hone in on this red bar, and this is Streptococcus mitis phage, SM1. So strep mitis, you guys are probably most familiar with strep mutans, is that right? Causes cavities. It's not too distant. It's a common mouth flora bacteria. Strep mitis. The phage is interesting because it's a lysogenic phage that encodes these two proteins called PBLA and PBLB, and they're known virulence factors for endocarditis. And probably a lot of you are familiar with the correlation with periodontal disease and endocarditis. If you have chronic inflammation, you're more susceptible. And so what this is just showing you is that this is just coverage of the phage genome, and that all of these blue little bars is showing you, how many sequences we found. But we found quite a few to PBL A and B. So my understanding of the strep and the endocarditis is that, it's bacterial endocarditis, so that's this infection of the heart tissue, right? Bacterial infection. It's very important, so attachment is like one of the most important things with this type of pathogen. If you can't attach to cells, then you can't grow there and cause all this disruption, right? In order for strep mitis to be able to cause endocarditis, it needs this prophage, SM1, and what it does is that it helps it bind to platelet cells. I'm going to show you. So these are the players, and then I'm going to have another picture where I'm going to show you how it works. So here's your strep mitis with the SM1 phage. Okay. It actually needs, in order to make it so that the strep mitis can bind to platelets, it actually has to be induced. So it isn't like the cholera where it's just sitting there in the genome. It actually needs to kind of be the induction process being started, because you need to start making these holins and lysins, which is part of the lytic cycle we talked about. That's the way that it's making the cell kind of compromised so you can blow it up. And it's kind of interesting because you're basically, if you're doing this, you're like a kamikaze cell for the population if it goes all the way to completion, right? If it doesn't go all the way, what it seems to happen is that first you make holins, which is like a pore that, that proteins can come out of, and lysins. So what seems like happens is you make holins and lysins. Once you have the holin pore, the lysin proteins can come out of that pore, and now they're in the system, right? They're outside of the cell. These lysin proteins are what makes the connection between the strep mitis and the platelet. Okay? And so now, because the lysins are just kind of in suspension, like outside of the bacterial cell, now you can get binding of lysin to the cell, the outside of the strep mitis, and to the binding of, this is fibronectin, part of the platelet. Now any old strep mitis can bind to it. It doesn't need to have the phage anymore. Okay? So this is like a population thing. So even though this one may ultimately get blown up, you're helping out your buddy here to bind to platelets. And also, the lysins also compromise the cell wall and allows PBL A and B proteins to come out. And those make kind of this bridge to, this is another like extracellular protein on the platelet. It's called GD3. And it also attaches strep mitis to the platelet. And so, and I don't know how that works. Like it could be that it doesn't really fully come to complete, I mean, this definitely damages the cell. And even if it allows you to bind to the platelet, it seems to be that PBL A and B actually need to bind to the surface of infected strep mitis. That's what the model is right now. It's binding to platelets. But the lysin part definitely is more free to bind other types of strep mitis to platelets, okay? So other guys in the population. All right, and so this we did as part of this study once we found the SM1 phage in the healthy human mouth, was to see what you could do to actually get it to induce, because you actually need that induction to complete that process where it causes the endocarditis. And so I'm showing you this again because when you induce the lysogen, you're basically putting it through the lytic cycle. So ultimately, you're going to get lysis and free virions, okay? And what I'm showing you in this graph is free particles. So we didn't count cells that were like infected with viruses. We literally just counted free virions. And so if you have, and this was a cultured system, so we knew that we had a strep mitis, which an SM1 phage, a temperate phage, SM1 inside. And then you can take this chemical, it's called mitomycin C. It's a known inducer of lysogenic phage. So you give it to the bacteria, you just add it to the tube, and you get tons of free phage particles. Okay? It worked. So then we tried to add all these foods that are certain, well, mostly drinks that people eat to see if they caused induction. Okay, and a couple of them did. So this nicotine, this is actually the liquid smoke that you'd like put in a vaporizer. It induces phage. This is just one phage. We don't even know of everything going on in your mouth, like what could be happening, right? And soy sauce, kind of surprising, okay? And so, I mean, it's kind of food for thought, right? It's interesting, right? So say you went to sushi, and then you had your vaporizer, and then you flossed your teeth, and you made it bleed. Now you've got potentially all of these phage particles that can cause disease into your blood system. That's just one story, right? Are you excited about phage? Okay. Don't be terrified. All right. And that's all I got. So this book was written by Forrest. Forrest has been my mentor for the last 15 years. And it's a very good book. It's meant for the lay public, right? It's got a lot of pictures, a lot of art. It's really interesting. It has a lot of stories. It's available to both buy and you can download it for free. It's very nice. And I will take questions. Thank you very much.
So we're looking for reasons why bacteria in root canals move from a dormant to an active phase, why we get flare-ups. So maybe this is one of the links that we're looking for.
Yeah, I mean, it could be. And Gary brought that up to me, and I've spent some time thinking about it. I don't know specifically how it could be caused by a dormant state off the top of my head, but these certainly, what I was trying to illustrate was they so strongly regulate bacterial populations and wreak all this havoc through those communities that they very well can be a huge part of many things that you're seeing, and especially changes that. So, I mean, is that when you're thinking that you think it's dormant, but you still probably are having some population changes that you're not seeing at the strain level.
So the phage has a significant water content?
No, they're all just nucleic acid and protein. There's some of them are enveloped, but and the membrane envelope would have some, but those are more eukaryotic.
Is there a high phase content in circulating blood? Just kind of a simple question. Would there be a high phage concentration in circulating blood?
There are some circulating viruses in your blood. A lot of them are human ones like EBV, and that seems to circulate in there a lot. We did do a blood study for phage. I mean, there are, your blood's not as sterile as you'd hope it would be, and there's some, but it's not a lot. It's not as much as everywhere else. In the same vein that there's not a lot of bacteria in there either.
So the phage, they can cross the blood-brain barrier, right?
I don't know. I think that they're still kind of subjected to the same tight junction things as. And they can't really be doing anything if they don't have a host. So they might be coming through if you got strep B or something.
So are the dynamics of the lytic temperate dynamic, do we know how that changes versus a dormant kind of biofilm versus planktonic rapidly multiplying organisms? I mean, what controls this transition?
I mean, I guess I would say that if you think your biofilm is growing slowly, you know, the classical thinking would be is that you'd have more lysogens in that system than lytic lifestyle phages, because you're seeing more swings in a lytic cycle. And so if everyone's kind of all glued together in the biofilm, they're all differentiated and growing kind of slow, then it's a better lifestyle to live lytically, where you grow, divide slowly with the host. And maybe if you left the biofilm, it would be a reason to be induced. You can have, and it would be an easy thing to look up and to test whether you had more lysogeny in a biofilm than lytic.
What controls it when you study this coral biofilm?
So I'm mostly, I focus on the water column above the coral, which does tend to change a lot based on who's there. So if you have, and in our system, we're seeing corals being overgrown by seaweed. And when you have seaweed, it, you know, you grow a lot more bacteria. Coral kind of keep it very happy and low level bacteria.
Research with looking at phages interacting with eukaryotic cells, maybe possibly as using them as anti-cancer or something.
Can you say it one more time?
Is there research using phages in eukaryotic systems, possibly as anti-cancer cells, if you had phages that you could direct to a particular?
Well, so in molecular biology, you can use a phage to do a lot, because you can use a phage as a vector to move a lot of genes. So you could, and it could be that they use phage to deliver anti-cancer things, for example. You're not technically, the phage wouldn't infect eukaryotic cells, but you can use them as tools.
You know, the whole CRISPR-Cas thing is using this model of how Phage works. That's how they're actually modifying the human genome. You can use this CRISPR-Cas method process to actually modify the human genome. So we have a speaker next year at the IAE. No, this year.
Next meeting.
Next meeting. It's going to talk about this whole CRISPR-Cas thing, which comes from this whole bacteriophage process. It's fascinating. So there's no question we're going to be fighting disease by modifying the human genome, using how bacteria protect themselves against this kind of bacteriophage.
Well, the monoclonal antibody theory that was out for, what, 20 years ago, when they were targeting specifically how to take down certain types of cancer, I think prostate cancer was being one of them. Is this now the next generation of thinking as you're now modifying this genome?
Well, I do think that people are trying to see, because phage are more inert to eukaryotic cells than other things that we use, certainly cytotoxic chemo drugs, right? And so if you can use them to selectively recognize a structure and destroy it somehow, then it's a good thing, because it doesn't. So, you know, we use phage therapy for many things, mostly outside of the body, like these chronic wounds. So nobody has a really big problem with using a spray bottle to spray phage on these wounds that are caused by bacteria on the outside of the cell. That's basically it, though, especially in this country. Nobody wants to feed somebody viruses to treat something that's on the inside. Everyone's pretty much okay with the outside thing. But it is their viruses, and you know, they move genetic material prolifically. I mean, you know, I think it freaks people out. Yeah, you could target MRSA with it. So phage therapy is bigger in like Eastern European countries. They use it more. Thank you.